Thousands of people are taking this peptide with Ozempic right now to completely wipe out the side effects. It repairs tendons, boosts new blood vessel growth, and literally shields your brain cells. But there are practically zero human clinical trials. And today I'm going to go through every level of evidence for BPC-157. And I will explain what we can say about BPC-157 with confidence and what remains unproven and pure speculation. Hello, my name is George and I'm a researcher's biologist. I've spent 15 years working in fundamental research and the last 6 in human clinical trials. Let's take a look to scientific publications. BPC stands for Body Protection Compound. The 157 in the name represents the specific sample number it was isolated from. Structurally, it is a peptide, a sequence of 15 amino acids. A peptide differs from a protein simply by its size. Peptide fewer than 40 amino acids and the protein more than 40 amino acids possessing a complex three-dimensional structure. You have probably come across similar synthetic peptides before. Ozempic and Wegovy contain semaglutide, a synthetic analog of a natural hormone from the small intestine called glucagon-like peptide 1 or GLP-1. BPC-157 was created using the same logic. It's synthetically made but based on a protein fragment found in the gastric juice. It was discovered in 1993 by a research group from Zagreb, led by Dr. Sikiric. They were looking for a gastroprotective proteins, but instead found a molecule with a much broader spectrum of action, but only on the paper. Let me explain why. In laboratory experiments on fibroblast cells from red Achilles tendons, it turned out that after treatment with BPC-157, the activity of FAK and paxillin signaling proteins responsible for the cell migration increased. The cells started migrating more actively, the tendon fragments began to heal better. In chicken brews, BPC-157 showed an improvement in angiogenesis. More blood vessels formed in the embryonic membrane treated with the peptide compared to the control group. The study even managed to map out part of the mechanism. BPC-157 increases the amount of the VEC4-2 receptor. It was exactly the interaction with this receptor that triggered intracellular signaling cascade influencing blood vessel growth. In rodents, BPC-157 demonstrated muscle mass preservation during extreme cancer-induced wasting. Furthermore, it turned out that BPC-157 normalizes dopaminergic and serotonergic transmission in nervous system. In rat and mouse models of Parkinson's disease and multiple sclerosis, administrating the peptide mitigating symptoms. When you first see these studies, BPC-157 looks like a miracle molecule, but the longer the list of effects for a single substance, the closer you need to look at who exactly proved it. We will come back to this point later. Most studies demonstrating the various effects of BPC-157 were conducted on animals, which means the basic principle of translational medicine applies here. We have already learned how to cure cancer in mice and Alzheimer's disease in mice too, but practically almost none of these models have successfully translated to human clinical trials. Drug metabolism and molecular targets differ far too much between our species and certain processes. That's why any molecule claiming to be useful for humans needs clinical trials. And this is where the real story begins. It's been 33 years since BPC-157 was discovered. From that time, exactly 2.5 clinical trials have been conducted. The first study on the BPC-157 peptide was conducted by the Croatian pharmaceutical company Pliva on healthy male volunteers to assess safety, traceability and pharmacokinetics, meaning what happens to the molecule when it enters the body. In this study, the peptide was administered via enemas. They concluded that the peptide is safe and well tolerated. However, phase 1 did not investigate efficiency. It only looked at toxicity and safety in healthy individuals. Efficiency was supposed to be evaluated in phase 2 on people with ulcerative colitis. But even though phase 2 of the study was completed, its results were never published. Another study on BPC-157 was conducted in 2015. Once again, it looked at safety and pharmacokinetics, and once again, the results were never published. This in itself is a suspicious signal. When a company finishes a trial and stays silent, there are usually two reasons. Either the patent rights expires, making it commercially pointless to publish the results, because other companies would use them without spending money on research, or the molecule didn't perform as expected. And so far, things aren't looking good for BPC-157. The only study that offered any kind of efficiency assessment involved administering the peptide for knee pain. This was a retrospective study, meaning patients received BPC-157 first and only then were they enrolled in the study and interviewed over the phone. Administering the peptide reduces the subjective sensation of knee pain in 14 out of 16 people. But the study lacked randomization and didn't measure anything besides the perception of knee pain. This is a highly subjective metric that depends heavily on how a person self-reports their pain. Now add another fact to the mix. More than a half of all scientific papers on BPC-157 have been published by a single group, that one from Zagreb. The exact same group that discovered the peptide and tried to monetize it through patents. This doesn't mean they falsified anything, but it does show a total lack of independent experimental replication where a different lab would confirm similar results for BPC-157. Fair enough. So where do we find reliable data when there's only 2 clinical trials and the preclinical data is questionable? We look at what's happening in real world. Science, plenty of people choose to take this molecule anyway. So I checked the FAERS registry, the FDA Adverse Event Reporting System. It tracks voluntary reports of adverse side effects from various substances. It's important to know that the registry does not establish direct causation. However, everyone who filed an official compliance with the FDA regarding BPC-157 faced severe consequences. Half of them were hospitalized. What stands out is exactly what these people were taking. BPC-157 doesn't appear as an isolated compound. In one case, it was combined with CGC-1295, growth hormone secretagogue. In others, it was mixed with thymosin beta-4 and sermorelin. The compatibility of these combinations has never been tested anywhere by anyone. Ozempic is a whole different story. Novo Nordisk officially petitioned the FDA to ban the pharmacy compounding of semaglutide. This is because some compounding pharmacies in the US were specifically selling blends of semaglutide and BPC-157. The logic was clear. Ozempic causes GI side effects and BPC-157 is a gastroprotective agent that should theoretically ease them. The assumption didn't pan out. The FDA flagged this combination as unsafe. This means getting your hands on BPC-157 is only possible via the gray market. And here's what that looks like in practice. In recent preprints, researchers analyzed 6,441 peptide samples. Between 41% and 71% of the samples failed quality criteria for either purity or composition. 15% of the sample in a smaller subset contained endotoxins, components of the outer membrane of gram-negative bacteria. If these endotoxins get into bloodstream, they can trigger allergic reactions and severe side effects. The thing is, bacteria are used to synthesize peptides in the laboratories. Then expensive chromatography is used to isolate the peptide, but apparently it's not being used everywhere and they are cutting corners on purification. Funny enough, but 2.4% of the samples didn't contain the advertised peptide at all. This is a systematic issue with unregulated manufacturing because there's no mechanism to enforce quality standards. Let's sum up the risks. Unknown interactions when mixed with other peptides. Unstudied long-term effects, a very real chance of getting a contaminated product or no actual product at all. BPC-157 is a molecule with interesting animal data, but the majority of the most promising studies were done by the very same scientific group who discovered it. The scales weighed heavily on the side of risk, and notice this. The risks here are far less speculative than the potential benefits. Because the FAERS compliance are real, and the contamination data is derived from actual physical samples. If I were to personally take the risk of ordering BPC-157, the very first thing I would do is send the batch to an accredited lab for a purity and composition analysis. Just a few people want to play Russian roulette with their own money. Only new clinical trials can change this picture. By the way, on clinicaltrials.gov, the first trial investigating the regenerative potential of BPC-157 for musculoskeletal injuries in humans have been registered. Interim data may drop in 2027. This will provide the first real clinical effects regarding its efficiency. Until that moment, any recommendations for BPC-157, including those from Andrew Huberman or other scientists, doctors, or biohackers, remain pure speculation. My video is strictly for informational purposes and does not constitute medical advice, simply because you cannot give advice when the evidence base is practically non-existent. Links to original paper can be found in my X, and you can support my scientific breakdowns on the Patreon. Click the links in the description of the video. Leave your question in the comments below, I always reply, and also feel free to suggest topics for future episodes. Thanks for watching.